The Evigence Sensors Laboratory is located in Nesher, a town in the north of Israel adjacent to Haifa. Occupying approximately 500 square meters, it's staffed by a team of engineers, chemists, and subject matter experts, many of whom are graduates of the nearby Technion, Haifa's world-renowned technical university. The team is led by senior Evigence executives, each of whom brings vast experience in their respective domains. In addition to the administration, sales, and logistics departments, the technical space is divided into 3 individual laboratories, each performing a distinct function, namely chemistry, kinetics, and quality control. Our process begins in the chemistry lab, in which we work on developing improvements of the existing layers that comprise our sensors and on developing new layers. The primary objectives include improving the performance of the sensors, specifically their accuracy, along with developing layers that enable additional time-temperature profiles. This is achieved by either influencing the rate of the chemical process or by changing sensitivity levels to temperature. In the laboratory, the R&D team prepares new compositions of adhesives and UV agents, conducts coating experiments on a variety of films that comprise the substrates of our multi-layer sensors, and performs drying and curing experiments under various controlled UV conditions. The chemistry lab is stocked with equipment required to prepare compositions in small quantities, but which are designed to represent large-scale industrial processes that are used in the production stages, including both coating and printing processes. The properties of new materials and coatings are then investigated further in our kinetics lab. Here, we store samples at a variety of temperatures in highly controlled incubators, depending on the time-temperature profiles that we are solving for. The incubators enable a very high degree of control in both temperature and relative humidity conditions that the samples are exposed to. Samples are stored at a number of different temperatures for varying lengths of time, from hours through months, and based on a predetermined protocol, they are removed for kinetic testing of the reactions, the rates of which are time and temperature dependent. Given that the progress of the reaction is expressed by a visual change, more specifically a change in color, the tests are of an optical nature whose objective is to document the color of the sensor and, importantly, the change in color as a function of time and temperature. The tests are performed by advanced optical equipment, specifically photodensitometers, that split the color readings into individual components, for example by using the LAB color space, in which L represents lightness of the color, A represents the location of the color on the red to green continuum, and B represents the color on the blue to yellow continuum. All readings are automatically loaded into spreadsheets that present the kinetic graph of the reaction over time. Flattening of the graph's curve indicates the end of the reaction, or as it relates to a product shelf life sensor, the end of the product's shelf life. In addition to the optical testing, the sensor's color change is also documented in controlled lighting conditions by a digital camera, providing a visual history of the progress of each reaction from start to finish. The third and final laboratory is where our quality control processes are performed. In this phase, our QC department is responsible for 2 distinct stages. Firstly, checking incoming material that will be used in the manufacturing of sensor layers, and secondly, checking final products in the form of complete sensors before they are dispatched to customers. In stage 1, the raw material is checked. This comprises of rolls of film substrates which are coated either with our proprietary active ingredient, the upper layer of the sensor, or with a metallized deposition that forms the lower layer of the sensor. The precise thickness of the aluminum deposition is crucial to the product and is inspected using a spectrophotometer. Note that both layers are produced by approved third-party manufacturing partners in accordance with well-defined product specifications and manufacturing protocols. Kinetic testing is performed on representative samples taken from these rolls, and those rolls that meet the required specification are released for production at our approved printing partners, where they are printed, slit, and packaged, where they wait to obtain release approval from QC in Stage 2. In this second QC stage, representative samples of the sensor layers that have already been printed as described previously are returned from the printers to our QC laboratory, where their kinetics are tested, and the samples also undergo a number of additional control points, including confirmation to size, printing quality, and release from liner tolerances. Only products that fall within our tightly defined performance tolerances are certified for shipment. Samples undergoing QC kinetic testing are stored in water baths, which offer tightly controlled temperature conditions. The samples, which are placed into sealed aluminum bags and immersed into the water, are removed for optical testing at predefined intervals. The water baths provide the highest possible consistency and accuracy of storage temperatures, and our equipment is suited to meet the stringent requirements of the World Health Organization, which administers quality control procedures for sensors used on human vaccines, which it sponsors and distributes globally. Finally, we are proud to carry a number of quality standard certificates, including the ISO 9001 Quality System Standard. All of our sensors also comply with EU and FDA standards for indirect food contact packaging. For more information related to our R&D or QC procedures, please contact either Dr. Inbar Dag, VP of R&D, or Noah Makhlouf, VP of Quality Assurance.